Hello everyone, and thank you for being with us today. In this 30-minute session, Dr. Bruno Buisson will be presenting how Neuroservices-Alliance can be a useful Swiss knife for your pain research program, as we have developed a disruptive assay platform for pain preclinical drug discovery. My name is Diane Royer. I am the marketing manager at Neuroservices-Alliance, and I will be moderating today's webinar. This webinar is number 8 in our CNS drug discovery series. You can find all of our webinars replays on our YouTube channel that I invite you to follow. Our webinars aim to provide attendees with strong insights on our various preclinical CNS drug discovery platforms and guide you in your assay choices. The presentation will last 20 minutes and will be followed by 10-minute questions and answers. If you have any question at any time of the presentation, please submit them in the question box that is located on the bottom of the GoToWebinar panel. I will receive your questions and read it to Dr. Buisson after his presentation. Also, please note that the session is recorded and you will receive the recording after the live session. Before starting, I would like to say a few words about Neuroservices-Alliance for those who don't know us. We are a consortium of CNS CROs specialized in vitro and in vivo electrophysiological and behavioral studies. Our goal is to deliver endpoints facilitating decision-making for our clients for their CNS drug discovery programs. We value direct and efficient interactions and rely on a team of more than 50 PhD scientists. As a speaker, we have Dr. Buisson with us today. He is the co-founder, president, and chief scientific officer of Neuroservices-Alliance. Hello, Bruno, and I will let you take the lead from here.
Hello, Diane. So I am very pleased to be with you today and so to introduce you to the preclinical pain discovery platform of Neuroservices-Alliance. Just as a reminder, so today most of the painkillers that were used in clinic have been developed more than 50 or 100 years ago. For example, if we consider morphine, acetylsalicylic acid, ibuprofen, or paracetamol. There is also a big challenge in the USA, so you know the opioid crisis. So there is a huge demand for non-opioid, non-addictive drugs to treat pain. And if we consider also neuropathic pain, there is a huge demand from the patients. So there is still the need for tools to develop new drug killers, painkillers. As a first slide, so I will just to move over a general presentation of the preclinical pain discovery platform of Neuroservices-Alliance, since we can propose, I would say, different type of solutions. For example, we can perform assays on different animal species, for example, rodents, dog or monkeys. And we have also the possibilities to provide assay based on human tissue, for example, native human sensory neuron or iPSC-derived sensory neuron. We can provide, I would say, cell-based assays using neuron cultures and especially human sensory or rodent sensory neuron cultures. We can provide cells-based, slice-based assays. And finally, we are able to provide some in vivo recordings in anesthetized rats or mice. For the human part, we can provide cell-based assays using human sensory neuron from donors or iPS-derived sensory neuron. And last point, we can provide some clinical consulting for people who are interested to have some insight from clinical experts for pain drug discovery. So I will move over the presentation by presenting the different type of assay, and I will start with the cell-based assays. As a first illustration, so this is a very classical model, so using rodent sensory neuron cultures. Those neuron in cultures could be recorded, for example, very classically with the patch clamp technique or the calcium imaging technique. Those sensory neurons could be taken from naive animals or neuropathic animals. And just as an illustration, so on this picture, you have a patch clamp electrode, of course, attached to a sensory neuron. And with the patch clamp technique, we can characterize, we can investigate different type of, for example, voltage-gated currents that are implicated in pain transmission or pain modulation. As an illustration on this panel, so you have N-type calcium current that could be isolated and recorded in a sensory rat sensory neuron. So with the open dots, so open circle, you have the profile of the IV current relationship for these sodium currents. And in the presence, for example, of very well-known drugs modulating this type of current, such as pregabalin or mirogabalin, we observe a very important and very sustained block of this current. And as a positive control, we can use omega-conotoxin that will cause a complete block of the N-type calcium current. On the right panel, you have an illustration of the characterization we can perform, for example, with neuropathic rat sensory neurons taken from, of course, Bennett model. And this is an illustration of the IV curve relationship we can determine. So in control condition with the orange trace, and in blue, which is the IV curve corresponding to the sodium current we record from neuropathic animal. And you can observe that the profile of these two curves are not similar and indicate that in the neuropathic situation, the properties of the sodium channel is different from the one of a naive sensory neuron. I will move to this second slide that introduces patch clamp recording performed in dog and cyno sensory neuron by the neuroservice team in the USA, so with Dong as a very skilled electrophysiologist. So an illustration of a patch clamp recording of sodium channels, sodium currents, so from dog, and we have an illustration of the effect of tetrodotoxin. And this illustrates that part of the sodium current is sensitive to tetrodotoxin, but it remains a large part of the sodium current, which is TTX resistant, and namely, of course, supported by, for example, NAV 1.7 or NAV 1.8 channels. On the bottom, this is another illustration of sodium channels recorded this time in CYNOMOLGUS sensory neuron, and also the illustration of the effect of a low concentration of tetrodotoxin, so provoking only a partial inhibition of the sodium current. Another illustration of what you can do on this sodium current, so you can evaluate compound modulating the sodium current in the, for example, CYNOMOLGUS sensory neuron. So you have in black, so there are the control traces, and at different holding potentials at -100 millivolt or -50 millivolt. And then subsequently, the traces that have been recorded in the presence of one compound. And you can see that this compound provokes only a fraction inhibition of the currents. And this inhibition is dependent from the holding voltage, since we observe a more pronounced inhibition at the holding voltage at -50 millivolt than at 100 millivolt. And as a reference pharmacological control in this assay, we use 100 micromolar tetracaine that triggers a complete inhibition of this sodium current in the CYNOMOLGUS sensory neuron. Then I will move to the assay we can propose using slice and spinal cord slices. Those slices could be prepared from rat or mouse, and they can be prepared from naive or neuropathic animals, so neuropathic animal, mainly the Bennett model. First, we can record these slices using the multi-electrode array, which is a technology dedicated more to neuronal network investigation. And as an illustration, for example, we can pick up the neuronal firing activity in the dorsal part of the spinal cord. And on this firing activity, of course, we can investigate the effect of compound and painkillers. The other possibility to record spinal cord is to use the patch clamp technique. And on this picture, you can see a patch clamp electrode placed in the upper layers, or maybe layer 1 or layer 2 of the spinal cord, so at the end of the sensory input. And we can stimulate the rootlet, so the sensory input, using a suction glass electrode that will, of course, artificially evoke responses, sensory response in the dorsal spinal cord. And on the right panel, you can see two types of evoke response we can record with, on the left, very monophasic and very simple response, whereas on the right side, you can observe a more complex and more polyphasic evoke response. Of value, both evoke response are blocked by CNQX, which indicates, of course, the glutamanergic natures of this sensory evoke response in the dorsal spinal cord. As, of course, the case studies, we can illustrate some kind of investigation we can perform using spinal cord slices. And as a first example, I would just illustrate a question we have had a long time ago from one customer that was interested in characterizing an activity of a positive allosteric modulator on native ionotropic glycinergic receptors. And I will illustrate how we have been able to answer to this customer's request. So what we have proposed is to perform patch clamp recording in rodent spinal cord slices and rat, especially spinal cord slices. So this is an illustration. So just to figure out where are located the glycinergic receptors in the upper part of the spinal cord. So you can see with this immunostaining that the glycinergic receptors are highly expressed in the dorsal spinal cord, very close to the sensory inputs. So what we did to make this characterization of the PAM is that we have made wholesale recording with the patch clamp technique, so with the patch clamp electrode, which is now just placed on the membrane in very close contact to the membrane to record all the currents of the neuron and to activate the ligand-gated, so the glycinergic receptor. So what do? We use a puffing electrode, and of course, this puffing electrode will be used to puff the glycine on the neuron and to elicit the glycinergic evoked current. The slice is placed in a large well where we can, of course, perfuse the well using the standard ACSF medium. And in this ACSF medium, of course, we can add the vehicle or any additional compound, such as the positive allosteric modulator. So if we move to the full protocol, so how it is organized. So at first, we will make some, I would say, control verification. So we will evoke glycine response using 20 micromolar glycine and to evoke, I would say, a basal evoke current. And we will repeat this very basal application a couple of minutes later to see that we can reproducibly and reliably evoke glycinergic currents. Once we have verified the stability of the glycinergic evoke currents, we will perfuse the positive allosteric modulator into the bus, and we will repeat the protocol of stimulation with glycines. And this time you can observe that when you make the puff of 20 micromolar glycine, we record a much larger amplitude of the glycine evoke current, of course, that reveal the pharmacological activity of this positive allosteric modulator. And last point, as a pharmacological verification and pharmacological control, we will add into the bus strychnine, which is a selective antagonist of glycinergic receptors. And in the presence of strychnine, we just observe that we have no more induced currents that demonstrate that the current that we recorded before was, of course, mainly evoked by the activation of glycinergic receptors. So on the left, on the right graph, you have the summary of the experiment. So with the control current that we have in the presence only of glycine, the control current in the presence of the PAM, and in this time we observe more than twice increase in the amplitude of the current. And the last point is the check and the verification of the nature of this current, and is full block by strychnine. Another illustration of assay we can perform using a spinal cord slice, for example, is screening small library of compound, and for example, to compare their activity with a very well-known reference compound such as morphine. And so we can illustrate this type of investigation, for example, using naive slice from RODENT and making some multi-electrode array recording. In this case, the principle of the assay is using a low concentration of capsaicin, so 100 nanomolar of capsaicin, to elicit a relatively steady firing activity in the dorsal spinal cord. And of course, on this steady firing activity of the neuron in the spinal cord, we can, of course, test painkiller compound and, of course, pain modulating compound. Here, this is an illustration of the effect of morphine in this assay, and you can observe that using two concentration of morphine, 0.1 and 10 micromolar, we observe a progressive and complete block of the firing activity, which is induced by 100 nanomolar capsaicin. Another illustration of a pain modulating compound in this assay is the effect of lidocaine, so local anesthetic. And you see that using, I would say, successive step and successive concentration of lidocaine, we will observe a progressive inhibition of the firing activity of the neuron of the dorsal spinal cord. So this type of assay is a very simple and very quick assay to screen painkillers compound on capsaicin-evoked induced activity. Now I will move to introduce our in vivo pain drug discovery platform, and namely using anesthetized rodents. So those rodents could be rat or mouse, and they could be either naive or neuropathic. In this type of assay, in the anesthetized animal, we will perform a dorsal laminectomy, just of course to access to the dorsal spinal cord. And once the dorsal spinal cord is accessible, we will place a very fine glass electrode in the dorsal spinal cord to be able to record extracellularly, evoke nociceptive or evoke pain response. And we can use any type of stimuli. We can perform some mechanical stimulation, some thermal stimulation, chemical or electrical stimulation. And while we are making this stimulation, we will record the evoke response for the, so for example, some burst firing activity in the dorsal spinal cord. And on this panel, you have an illustration of different type of response that could be evoked, for example, by pinch, by water jet at 50 degrees Celsius, or using, for example, mustard. And you see that the profile of the response are very different depending from the stimuli that you use. And of course, this type of assay is very well dedicated to assess the activity of your painkiller compound, and the compound could be administered intravenously, IP, or orally. And just as an illustration of what we can do in this vivo assay is the illustration of activity of a TRPV1 antagonist, namely BCTC. So in those experiments performed in anesthetized rodents, so we have used different stimuli. So the first stimuli is a mechanical stimuli with a Fonfrey filament. And what you can see is that when you make a mechanical stimuli, BCTC is without activity. It does not change the evoke response by the Fonfrey filament. Whereas if you apply a thermal stimuli, for example, water jet at 40 degrees Celsius or water jet at 49 degrees Celsius, we observe that the presence of BCTC that has been injected intravenously in the animal will completely inhibit the response. Of course, that illustrates that for noxiostimuli and thermal noxiostimuli, TRPV1 receptors are implicated. Then I will move maybe to the most interesting part for drug discovery is the use of human material to assess the pharmacological activity of compounds. So within the Neuroservices-Alliance, we have a platform called ANABIOS. ANABIOS established a unique partnership with many hospitals in the USA where they can access to DRG from human donors. And of course, those DRG in cultures could be used like the rodent DRG to investigate many targets like voltage-gated channel, TRP channels, glutamate receptors, mGlu receptors, etc., etc. Those compounds and those targets could be investigated with two technologies. The first one is the calcium imaging, and the second one is the patch clamp. With calcium imaging, so this provides the opportunity to record in parallel many sensory neurons, of course, and to benefit of response of a population of sensory neurons. Since those neurons are relatively quiet or very silent at rest, we apply an electrical stimulation, of course, to trigger evoke response in those sensory neurons. And what you can see on the bottom with the red line and the blue line, those are, I would say, evoke an increase of intracellular calcium in those neurons triggered by the electrical stimulation, and with two different profiles. One where you have, I would say, cumulative, an increase of the cumulative response of intracellular calcium, whereas for the second neuron, you observe a more steady and somehow desensitization of the evoke response. On the right panel, you have illustration of I would say evoke response using current injection at different frequency, and you can by this way trigger action potentials in those sensory neurons at different frequency. And of course, on these evoke action potentials, this provides the opportunity to test compound. And you can see on the bottom illustration, so this is the activity of compound A that will modify the profile and certainly the firing activity of those human sensory neurons. Another possibility to investigate the drug properties on human material is to use human iPS-derived sensory neuron, and this is one capability provided by Neuroservices USA. to investigate membrane properties, voltage-gated ion channel, ligand-gated ion channel, or GPCR modulation of ion channel using iPS-derived sensory neuron. And on the pictures on the bottom, you can see a low magnification or large magnification cultures of human-derived iPS sensory neuron at different number of days in vitro. And on the top panel, you can see voltage-gated sodium current that has been recorded in those iPS-derived sensory neurons. And for example, Dong has documented together with Bob the expression and the peak sodium current as a function of day in vitro. And you can see that after, I would say, a couple of days in vitro, we reach somehow a steady state of, I would say, peak sodium current amplitude that indicate that the neuron in cultures after maybe 2 or 3 weeks have reached a certain maturity, and there is a steady state expression of the sodium current. But for sure, those neurons are excitable cells and provide some properties of human sensory neuron for sure. Another possibility to record the human iPS-derived sensory neuron is to use high-density CMOS multi-electrode array, and we have established a partnership with Maxwell Biosystem to use their beautiful multi-electrode array system. And using this HD density multi-electrode array, we can also record in parallel many iPS neurons in parallel. And this is presented in the picture on the bottom, so it looks like star on a dark blue sky. And so I would say the bright points correspond to firing neuron in the well. And so you can notice that you can record many neurons in parallel. And of course, which is of interest is the possibility to assess and to evaluate compound activity on this firing activity. And this is illustrated on the bottom panel by the retigabine application in the medium. And what we can observe is that with increasing concentration of retigabine, we will progressively inhibit the firing activity of those human iPS-derived sensory neurons in cultures until to a complete and full inhibition at 10 micromolar retigabine. And last parameter we can monitor is the burst shape profile of those neurons firing. And of course, depending from the drug activity and the target, we can observe some modification in the burst shape, and that could give indication about the target and the mechanism of action of your compound. Jen, to illustrate what we can provide in more complex, I would say, protocols. So I will just give the illustration of lead compound that has been developed for pain. And if people have developed a lead compound using most of the time rodent tissue or rodent cultures, or even making some in vivo assay using rodent, there's still the question to be answered if those compounds have activity on human sensory neuron, of course, to provide the best transferability to clinical trials. So in this case, what we can provide, sorry, I will move to the right place. So we can provide multi-electrode array investigations of naive cells and human cells. Either it could be human sensory neuron, thanks to anabios, or it could be human iPS-derived sensory neuron. We can investigate, I would say, the compound activity while recording the firing activity of those neurons using the high-density multi-electrode array, of course, to have a clear pharmacological illustration of your compound activity on human sensory neuron. Once we have, of course, determined this activity, we can go one step further, making patch clamp recording, of course, to have a higher resolution and to investigate more at the cell level and down to the single channel, the activity of your compound on the target. And of course, in parallel to the multi-electrode array, we can perform some calcium imaging in both the human sensory neuron and the iPS sensory neuron, of course, to document the activity of the compound or more on a population scale level. The last illustration I will provide how to can use the Neuroservices-Alliance preclinical platform is, for example, when you have compound that have been empirically developed, and this is the case for many times, for example, for neuropathic pain compound. This compound has been developed empirically. They have made very nice proof of concept in the animal, but the target and the mechanisms of action are still unknown. And then what could we propose in this case? So in a first, I would say, approach, we can investigate the pharmacological activity and to pick up some kind of electrophysiological signature of the compound using rodent slices from naive animals. We can investigate the signature of the compound using the multi-electrode array, of course, to look at the network level if we observe some modification. And once we have picked up an electrophysiological signatures, we can use the patch clamp technique to further investigate the property of the compound on the target and the modulation at the target level. Of course, once this investigation have this performed, we have the possibility to repeat them again, but using slice prepared from neuropathic animals. So in this case, we will test the compound, I would say, on the diseased tissue. And of course, this is one more step to assess their efficacy and their potency on a neuropathic tissue. Then next step could be to investigate and to demonstrate the activity of the compound while making some in vivo recording. And we can perform some in vivo recording in neuropathic animal and to make a very nice illustration of the compound activity and making some electrophysiological recording to have a very clear-cut activity of the compound in a whole animal. And last point, of course, and to be, of course, more comfortable to move to clinical investigation, we can check about your compound activity using the patch clamp technique, but this time using human sensory neuron. Could be native or I would say neuropathic-like sensory neuron. And we can perform this also using a human iPSC-derived sensory neuron, of course, to completely validate the target engagement on human tissue. Then if we can summarize the preclinical pain drug discovery platform of Neuroservices-Alliance, please keep in mind that we can use different species, not only RODENT. We can perform recordings on dog, on non-human primate, and even on human sensory neuron. And we can combine, I would say, these different type of human sensory neuron and rodent and monkey sensory neuron. And we can investigate them with different technology. So namely the patch clamp technique. We can use HD multi-electrode array. We can use fine glass electrode for in vivo recording, and we can use intracellular calcium imaging. So this provides the opportunity to combine many types of cells, many types, of course, of assays and technology. And finally, also, please keep in mind that we can perform investigation using either naive tissue from naive animals, but we can use also tissue or cell culture derived from neuropathic animals. And the last point, remind that we can provide the in vivo recording in anesthetized rodent, and maybe this is the unique opportunity for you to have a proof of concept of your compound activity in a whole animal. It could be a whole naive animal or a whole neuropathic animal. So thank you for listening. I'm very happy to answer now if you have any question on this topic.
Thank you very much, Bruno, for this very comprehensive presentation. Actually, we do have a couple questions already. Have you seen the activity of NMDA receptor painkillers in some of your assays?
Yeah, this is a very interesting question, since the NMDA receptor has been investigated by many programs and many companies as a preferential target in pain drug discovery. So at Neuroservices-Alliance, in a couple of years ago, we have investigated the effect of memantine, because there is some clinical investigation that have documented, for example, that in preoperative part or even in postoperative situation, the use of memantine can prevent and can strongly reduce pain in mastectomy surgeries. And so there is a good clinical evidence for the activity of memantine to prevent pain. And we have investigated the effect of memantine on the spinal cord and on the spinal cord of neuropathic rats. But so far, we have not been able to document any activity of memantine in this assay. So this doesn't mean that NMDA receptors and NMDA modulators are, of course, devoid of interest, but we have not been able to translate some clinical observation into, I would say, preclinical modulation or pharmacological investigation using rodent brain slices.
Thank you, Bruno. Now, can you discriminate between different types of sodium channels in your assays, namely between NAV 1.7 and NAV 1.8 and others?
This is a frequent question we have because many pain program has focused on those targets, so namely NAV 1.7 and NAV 1.8. Of course, these channels are not expressed by some subpopulation or isolated subpopulation of sensory neuron. Most of the time, there is, I would say, a mixed expression of different type of sodium channels. So in this case, what we can provide is that we can, for example, of course, block the TTX sensitive sodium channels. But even with the TTX sensitive sodium channel blocks, the remaining TTX resistant current will be, I would say, a mixture of sodium currents passing through NAV 1.7 and NAV 1.8 channels. And if we need to make some discrimination, we will need to use some pharmacological selective tools that will be able to selectively block either the NAV 1.7, NAV 1.8, or NAV 1.9 sodium channel. So we have to combine and to use different tools to isolate the component of interest.
Thanks, Bruno. Do you have any experience with analgesics that affect the bradykinin receptors?
So far, no, we have not experienced. So this is a question of value because we know that, for example, when you are in neuropathic situations and what it is used, for example, to artificially place the sensory neuron in a neuropathic or inflammatory situation. So we use bradykinin or prostaglandin E2 to trigger, to artificially trigger, I would say, an inflammatory situations. So for sure, if you interfere with the bradykinin receptor, you will certainly have an effect on the inflammatory, on the neuropathic situation. But so far, we have not an experience using bradykinin modulators.
Thanks. What is the usual turnaround time between signature of work order to completion of work and delivery of data? For example, for a study in neuropathic rats or human DRG with 3 doses of compound.
So if we start with the neuropathic animal recording, so of course there is a delay which is related to the generation of the neuropathic animal because of course the surgery will be performed once the animal are about 2 or 3 weeks old, and to have the complete neuropathic situation established, this will take an additional 10 or 15 days. So there is roughly a big month to get the neuropathic animal, and once we have the neuropathic animal, so we will be able to perform either the multi-electrode array or patch lamp recording. So most of the time we can complete, I would say, a multi-electrode array or patch lamp investigation, I would say, within 1 month. So if you consider neuropathic animal and in vivo electrophysiology with multi-electrode array and patch lamp, you can consider this is about 2 months study investigations. For the human materials, I think it could be faster because, for example, at ANABIOS, they can access to DRG from human donors on a weekly basis. And at Neuroservice USA, we grow regularly human iPS-derived sensory neuron. Of course, there is the delay to wait for the neuron maturity, which is a question of weeks. So, but you can consider that waiting 2 weeks to get the iPS sensory neuron matures, and then a couple of weeks to perform the pharmacological investigation of your compound. So this is, I would say, a question of weeks, or I would say a few months for those 2 types of investigation.
Do you use spontaneous activity as an in vivo readout of pharmacological effect of IV dosed compounds?
So if we are talking of in vivo investigation, so in anesthetized rodents, of course, most of the time, the firing activity is silent in the dorsal spinal cord. Maybe with the exception if you work with neuropathic animal, because you know that in the case of neuropathic animal, you can have some ectopic activity. This is some kind of spontaneous firing activity reaching the spinal cord. But in naive animals, we need to make some artificial stimulation, so using mechanical stimuli, thermal stimuli, or chemical stimuli or electrical stimuli to be able to record evoked response in the dorsal spinal cord.
Okay. What do you mean with human neuropathic like DRG neurons and human iPSCs?
So if we consider the human sensory neuron derived from donor, so it's very rare and very difficult to access to DRG from neuropathic patients. So in this case, one possibility to work on human sensory neuron in neuropathic situation is to grow the human sensory neuron in an inflammatory medium, including, for example, bradykinin or prostaglandin E2, that will, I would say, artificially trigger an inflammatory situation and put the neuron, I would say, in a neuropathic situation, an artificial neuropathic situation. And we can use the same recipe for the IPS sensory neuron because, of course, we cannot access to IPS sensory neuron from neuropathic donors. So we have to work with, I would say, standard human sensory neuron. And then one possibility to have neuropathic human iPS-derived sensory neuron is to culture them in an artificial medium, including, for example, bradykinin and prostaglandin E2, to artificially trigger the neuropathic situation.
Thanks. Now, does ANABIOS have access to neuropathic pain patient DRGs?
Yes, this is what I've indicated. So ANABIOS can access to some neuropathic human DRG, but this is a very rare situation. So I think ANABIOS reserve its access to neuropathic donors DRG to, I would say, very particular and maybe high-level investigation with big pharmaceutical company, because since this tissue is very rare, of course, it could not be proposed to everyone and to any kind of company. So maybe the best option to access to neuropathic sensory neuron is to consider recording sensory neuron cultured in an inflammatory medium.
And do you confirm development of neuropathic pain in your Bennett rats prior to collect DRG?
Yeah, this is a good question. So for sure, we assess the neuropathic situation of the Bennett animals before using them. So the hyperalgesia or the allodynia of these animals will be checked, and the basic assay is that we will make a Fonfrey filament testing just to assess that those animals are really in a neuropathic situation. So before taking out the tissue, before taking out the DRG and the spinal cord. So of course we check, and we will use only confirmed neuropathic rat for those investigations.
Now, how much data from your experiments is sufficient for the MEA to register an innovative drug regarding mechanism of action? And also, at the same time, do you have any experience of MOA investigation of anti-Parkinsonian drugs?
Most of the time, so if we are considering the multi-electrode array technology, we can screen, say, compound over a couple of slices. So a couple of slices means between 6 and 12 slices per compound. There is also the possibility to make some cumulative concentration steps on the same slice. Of course, that we will save time and compound. Of course, this is relatively quick, and of course, the throughput is much higher than so what we can propose, for example, with the patch clamp technique. So the multi-electrode array technology, and for example, using either a spinal cord slice or brain slices, of course, is very well dedicated to screen a small library of compound because we can generate the data in a couple of weeks. Of course, for Parkinson's disease compound, we can, of course, use the same approach. But of course, we will not test the compound maybe on the spinal cord, but more of brain regions and of interest, for example, maybe the substantia nigra or I would say the striatum. But the approach is the same one, so we will screen the compound over a couple of slices. And for example, if we consider only the buffering activity as a readout, this could be relatively quick to generate the data.
Thank you, Bruno. And I think last question probably. Are rodent models robust enough to have a good confidence in translation to clinic of preclinical pharmacological data?
I think the rodent model we have used so far are robust in terms of physiology, of pharmacology, but still remain the questions, even if we have been able to make a very nice and very precise characterization of the pharmacological signature of the compound using some rodent tissue or whole rodent investigations. The questions remain about the activity of the compound on human proteins. So even if we can provide, I would say, very robust and very demonstrative data using rodent tissue, I think it will be very cautious to make a check of the compound pharmacological activity using either human sensory neuron or human iPS-derived sensory neuron, because those investigations could be relatively quick and not so expensive. And this will provide you with, of course, the whole confidence that your compound have a good activity on the human protein. And sometimes, and this is the reason maybe some pain program have failed so far, is that because most of the data have been generated with rodent, and the difference in the protein between the rodent and the human was somehow responsible of the lack of compound activity while moving to clinical investigation. So we strongly encourage our customers, our partners, to check about the pharmacological activity of their compound on human sensory neuron or human iPS-derived sensory neuron.
Well, thank you very much, Bruno, for answering all the questions. We don't have any additional questions as of now. Thank you for your very comprehensive presentation.
Thank you.
The webinar has now come to an end, so you will soon receive an email with the full recording of this session and also the opportunity to follow up and to ask additional questions if you want to. Please visit Neuroservices-Alliance.com to make sure you register for our next webinars. And also, if you can fill the little survey that will show up in your browser after closing this window, we really appreciate your feedback. Thank you, everyone, and we look forward to seeing you at our next online events.